All right. Looks like we're at 10:01, so let's go ahead and get going. So today we'll have Hui, who is from Sanson's lab, who will actually be giving us a talk about a comparative study of CRISPR-based activation in maize. So with that, Hui, would you like to
Yeah, sure.
get going for us?
Sure. I will start. Mm, Thank you for the introduction. Hi everyone, my name is Hui Liu. I'm the postdoc fellow in Sanjin Liu's lab. So today I'm going to talk about a comparative study of approaches for gene activation and CRISPR screening in maize. So my topic contains the following part. So first of all, I will talk a little bit about what is gene regulation and how gene regulation influence plant development and fine-tunes growth and plant defense to disease, and how designed transcriptional regulators activate the gene
Uh, we
expression.
can't we can't
Uh,
see your screen.
oh, sorry. Um, Sorry. Share. Can you see me now?
Yeah.
Oh, my bad. So, I will summarize three methods here we are using, including the dTOR method and the CRISPR-based decasenet activation method called CRISPRa method, and canonical overexpression method. Here is the basic knowledge about transcriptional regulation. So, transcription begins with the translation vectors and the cofactors from pre-inhibition complex with RNA polymerase at the promoter region, and stabilized by the mediator and cohesion. So, polymerase II initiates transcription but soon pause downstream, and elongation. Elongations can start after the elongation factors release the pulse, enabling the transcription elongation. And also, chromatin rebonding factors open the nucleosome, while histone writers and erasers and readers regulate activity through modification of the histone proteins. Gene regulation plays important roles during the plant evolution process. So here I will give you an example about TB1. TB1 is a star gene, is a central regulator of maize architecture. In T20, TB1 expression is pretty low, and TB1 is a negative regulators for lateral branching and bushy form. And in maize, in modern maize, TB1 allele is expressed very high level, suppressing the side branches and promoting single and upright stalk with a large ear, which is an important step during the maize domestications. So what will happen if we overexpression the evolutionary genes? So here, if we overexpression TB1, maize TB1 in wheat, the tillering phenotype of the wheat will disappear. However, if we overexpress another domestication gene called TIN1B37, it will abolish the single stalk phenotype into the T20 phenotype. And another case is IPA1, which is also a star gene cloned from the rice. IPA1 is known to regulate the panicle branch and tillering by activating DEP1 and TB1. Also, IPA1 can enhance plant disease resistance by activating WORKI45. So, however, if you overexpression IPA1, the IPA1 expression will significantly increased. However, the yield will be compromised, but the defense response will increase. If we're using the conditional controlled promoter to drive IPA1 expression, make IPA1 expression level at moderate levels, the yield and defense will increase. However, after the XOO invasion, this is a password, and the IPA1 expression level will further increase without yield compromise and increase the defense response to disease. That is how regulate a gene to balance the trade-off between the yield and disease resistant implant. Apart from using the canonical overexpression method or conditional promoter method, designed transcriptional regulator give us another opportunity to modulate or manipulate gene expression. Here, synthetic transition activators are fused proteins between DNA binding proteins and activators. So DNA binding proteins. So before CRISPR technology came out, zinc finger proteins and tail proteins are widely used for engineering those synthetic transcriptional regulators. So DNA binding domain are designed to target any user-defined sequence of a genome in the promoter region. And chimeric activators can recruit transition factors in addition to the RNA polymerase and then to activate downstream gene expression. Tau protein is actually a natural transcription activator, has its own activator, but we still can modify these activators using different fused proteins. DCAS9 is a catalytic dead Cas9 protein. Through fused the DCAS9 with different activators, including VP64, P65, or EDLL domain can be activated the downstream gene expression after binding the get RNA to the target region. But the first two methods is based on designing a protein, while CRISPR is based on designing a get RNA. So here in our study, we are using the detail method and the CRISPRa method. Detail method is based on the tau effector itself, the activation domain. So we deliver the engineered the tau effector through the bacteria XVV. The tau effector can inject into the plant cell, and after binding with its target, using its own activator, which can activate the downstream gene expression. CRISPRa is much more easier for manipulating, only designing a get RNA here. Here is the pipeline of these three methods. Detail method, as I mentioned before, we are using the sensor models as a mediator to deliver our activator component. We inoculate the maize seedlings use the synergen to transfer this component. After 1 day's inoculation, we ground the leaf tissue and perform qPCR and RNA-seq analysis. Here, we're also using the protoplast system as a transient assay platform to deliver CRISPR-A vectors and overexpression vectors, and then we perform qPCR and RNA-seq analysis. So why we do this experiment? Because we know that modulating gene expression is very important for understanding biological process and synthetic biology. Here we developed two transient systems using three methods for gene activation. And our ultimate goal is to identify the best strategy for target gene activation in maize. For our case study, we are using GLAS gene as our case study. GLAS genes are involved in the biosynthesis of cuticular wax. So GLAS mutant, the epidermal cell of the GLAS mutant showing like this wax deficient phenotype. That's why you can see this glassy phenotype. GLAS3 is a MIP transcription factor. This gene was cloned by Sanzhen almost 15 years ago. And So Sensomonas persevere can inhabit the plant apoplast or xylem vacuoles and ejaculate the effector protein into the plant cell using the tapestry secretion system. The tau effector can translocate from the cytoplasm into the nuclei and then manipulate the host genome to transcribe the beneficial mRNAs for their spreading or for their propagation. So Sensomonas is very smart here. And what we can do is like we can modify the tau effectors to regulate the gene of interest expression. So tau effectors contains several tandem repeat. Each repeat can recognize one nucleotide. Through engineering different repeat, we can design any kind of tau effector here. So for our case study, GLAS3 was used here, and we designed two tau effector, and we checked the GLAS3 gene expression level after 24-hour inoculation using the engineered tau effector, and we also performed the qPCR and RNA-seq analysis. And interestingly, we found that these two tau effectors expression pattern showing good correlation. And interestingly, we found that 96 gene can be up-regulated by these two tau effectors, and 54 genes can be down-regulated by after a GLAS3 gene activation. And Go enrichment analysis showing that facet acid biosynthesis pathway and ER pathway has been enriched. That makes sense because GLAS3 contributes to the wax synthesis. These two pathways are very important for that pathway. And interestingly, we found that 6 already cloned GLAS gene are upregulated, and including the GLAS13, GLAS6, and GLAS1, 26, GLAS4, and GLAS2. And we also found several unknown GLAS gene can be activated by GLAS3 gene expression. So this work has been published by my former colleagues in plant cell in 2023. And so my research mainly focus on the CRISPRa method. Here again, we're using GLAS3 as our case study, and I designed two different get array. At beginning, we actually tested several CRISPRa system, and the TV system works pretty well for us. And we found that different get array can activate the native gene expression, and we also found that a combination of two get array outperform the individual get arrays. And we also tested the different time series after transformation. We found that 16 hours of inoculations is better than 48 hours inoculations after the protoplast transformation, mainly because of the degradation of the CRISPRa components. And we also tested the impact of OE GLAS3 using a strong promoter ubiquitin here. We are using here from Aurisa sativa. And again, we're using protoplast system, and fluorescence signal indicator we have a decent transformation efficiency. Around 50 to 80% of the protoplast can be transformed with our plasmid. And we perform qPCR. GLAS3 can be, highly induced by the strong promoter here. And we also compared these two methods, OE method and CRISPRa method. We found that these two methods both showing a comparable fold change of GLAS3. Again, we performed the RNA-seq analysis, and the x-axis represents the CRISPRa GLAS3 with the two get arrays to the empty vector, while x-axis represents the fold change of ubiquitin GLAS3 to the empty vectors. Both methods could highly induce the GLAS3 gene expression. Here, GLAS3 is the most highest expressed gene, like 2 power 15 is a huge induction of GLAS3. And these two methods showing good correlation. as you can see here. And we also found that most of already known GLAS gene can be upregulated by GLAS3 gene expression. And we compared our RNA-seq data from these three methods. And interestingly, we found that already known GLAS gene can be, simultaneously identified by the two in vivo method, detail method and CRISPRa GLAS3 method. However, actual OE GLAS3 cannot identify GLAS2 and GLAS14 expression. Nevertheless, OE method is the most robust method for drive mRNA expression. But the underlying reason we still don't know. But when we go back to check our former RNA-seq data from the plant cell paper, we found that GLAS3 expression of GLAS2 and GLAS14 associated with GLAS3 expression level, because in our former data, DT1 and DT2 showing different impact for treatment for GLAS3 expression level. So DT1 treatment of GLAS3 is higher than DT2 treatment, and the GLAS2 and GLAS14 can be significantly detected in the DT1 treatment, but not in the DT2 treatment. And to further demonstrate that how GLAS3 regulates the GLAS gene expression, we perform the CAR-Tag analysis, kind of like a ChIP-seq analysis, but CAR-Tag is much more easier to do, because we are using the transient system, and the input of the cell for enrichment only need 5,000 of protoplast. Again, we're using the transient system, and we designed the GLAS3 fuse with EGFP, and the EGFP itself serves as the negative control here. And we did 2 wrap, and we found that GLAS3 indeed can enrichment surrounding the GLAS4 gene body region and the promoter region. We further using the luciferase experiment to demonstrate that GLAS3 indeed can bend the promoter region of GLAS4. And for first demonstrate this CRISPRa method, we test several transient factors, including WUSHO1. WUSHO1 is a positive regulator for the stem cell development, but so as a repressor, transfer repressor. Here we designed 2 gather, and we perform RNA-seq analysis, and these 2 gather showing good correlation. So, and interestingly, we found that the differentially expression mainly clustered in the downregulated part, including 234 genes, and however 42 genes can be upregulated by CRISPRa WUSHO1, which is consistent with the former report about the WUSHO1 function. Here's a summary of these 3 approaches. So, strong promoter and detail method are based on protein design. CRISPRa is based on GalRNA design. So, compared with the former 2 methods, CRISPRa is much more easier to manipulate and easily to go multiplexing experiment. But strong promoter is more like exogenous induction method. Detail and CRISPRa is based on endogenous activation of the native gene. And among these 3 methods, for sure, strong promoter is the most robust expression method. But in our data, we found that CRISPRa can reach the similar or comparable for change of gene activation here. The reason why we're trying to find the optimal way to study gene regulation, because our lab has been working on a project called regeneration project for many years, and we generate a lot of candidate genes that can be contributed for potentially contribute for callus induction. So far, we already got a lot of data and found like at least thousands of genes can contribute this process. So if we are using the canonical method or traditional method to test our candidate gene, clone them one by one and test their function, it's almost we cannot do that. If we have only hundreds of candidates, it's doable. We can clone one by one and test their impact. But for thousands genes, it's time consuming and money budget is another issue. So here we are using CRISPRa method for our high throughput screening. Our design is based on B73 genome. For each candidate gene, we design 3 get array and we compare it. We construct a get array library and we then deliver our CRISPRa components into the B73 base using the gene gun shooting method to transform the B73 gene, B73 immature embryos. Here, why we're using B73? B73 is the most hard or recalcitrant lines to be induced for callus induction, nevertheless, than the regeneration of the whole plant. Luckily, as our preliminary data, we screened almost 200 of candidate genes. Luckily, we found that B73 immature embryos can generate this type 2 callus after the CRISPR treatment, and some the type 2 callus can regenerate the whole plant. So far, we already got several candidate genes, so this project is still going on, and I hope next year we can give some update of this project. And I'd also like to introduce Home tool tool for our colleagues, because last year we released this Home tool online, and which is very useful for comparing different genome to find the best homologous gene using different reference. Here, what you can do is you only need to provide a gene ID, let's say B73 gene ID, and searching the best homologs in the different genome, let's say here, for example, A1A. So what we can do, we can provide you the polymorphism between the different genome, and including the SNP, indel, and structural variations. We also can provide you the annotations of the different polymorphisms and what kind of impact of the different polymorphisms, and determine which kind of structural variations are associated with transposons. So please scan the QR and exploring your gene of interest to find more polymorphisms. And we would like to hear any feedback from you. And here is the output of using Home tool to search of gene candidate, gene of interest. So we will provide you the visualization of identity of different genes. We will provide you the genome variants with different impact, from high to moderate or even no impact. And we also provide you the structural variation. Please check it out. And I'd like to thank my colleagues at K-State, especially my supervisor, Sanjin Liu, and my lab members, Ravi, Lydia, Ala, Jean, Tengfei Liu, and Jinchen Zhao. I'd like to thank our collaborators, Frank Watt from University of Florida, and Jin Yang from UNL, Jun Zheng from CAS, and K-State collaborators, Xingang Park, Aaron, and Dr. Harold Trick. I'd like to thank NSF, USDA, and Energy DOE for their generous support on this project. And I will stop here, and I'm very happy to take any suggestions and questions for this ongoing project. Thank you all.
Thank you, Hui. So let's see. Really interesting talk. Let's just have a look through the questions here. I had like a kind of meta one. I'm not
Uh,
sure if anybody else would want to jump in and go
um,
first, but I can ask mine if there aren't any other volunteers.
so I can read through the
Yeah, like
online environment hormone is using the whole genome. A question from Dr. Jeffrey. I think Sanjun is the best person to answer this question because he's responsible for the developing of this tool. Maybe he's the better one to answer this question. Thank you,
Yeah,
Sanjun.
Jeff question is, did we do the whole genome alignment? We actually using a gene as a query, and the gene could be the gene region or extend outside region, and then doing alignment search to get the best target, and then perform the structure comparison, identify polymorphism, and notate the polymorphism. And yeah, it's pretty handy. I mean, there are multiple modules in the package, and home Maize will integrate, I mean, information and display in the HTML. You can easily scan. If anyone has a gene number, we can test it here. You can input it here, and then we can test online. I think yeah, we like the tool. It's not like a um revolutionary tool, but it's a handy tool to get information and save time. That's our purpose.
Have you thought about talking to the folks at Maize GDB to incorporate it into Maize GDB?
Yeah, that would be great. Yeah, we are not good to follow up design, and I think if MageD can host that, that would be great. And good suggestion, Jeff.
well, I guess I kind of had sort of a meta idea. So like more and more like when I hear about like these sort of comparisons between like transcriptomics and proteomics, like the data never quite matches like one to one. And as I was kind of thinking about one of the problems theoretically with CRISPR is that you have these sort of like off targets if you have anything that's like super
Yeah.
common. And in a
Yes.
way, could you actually exploit that? And like, if there are certain motifs that certain transcription factors might like, could you actually sort of use that as like a negative image to sort of compare against like the epigenetic landscape? So you would actually kind of know like where your histone marks or where your sort of like bisulfite marks might be in the genome. And you could then sort of compare that with sort of giving CRISPR the motif that you think your transcription factor might like and seeing how many of those it can actually kind of get into. Um, Probably not the way that you're thinking to use this tool at all, but it just kind of had me thinking.
So, thank you, John. It's a really good question. First of all, our the off-target issue actually we can avoid by improving our get RNA designing, and indeed we found that comparing the transcriptome using different get RNA, we saw a good correlation. The different get RNA showing similar impact and similar gene expression pattern. Maybe that can answer the off-target issue, and compare with the traditional CRISPR editing method. I think not all of the get RNA can target the gene even for the activate the gene expression. So this ratio I think is pretty low. And another question for using the different CRISPR fused with different modeling protein, right? Am I correct? Or I think another group already using modified engineered the decassenator using different modular chromatin modelers. This has been already done, but here we didn't test. And our goal is trying to find a good method to activate the native gene expression. To play with the gene which contribute to the topitancy in maize, especially for regeneration project. That's our goal. But for sure, we can play a lot using the CRISPR technology for modulating gene expression.
Well, thank you.
Yeah.
Looks like we just got a question from Jonathan in the chat. Jonathan, do you want to hop on and
Yeah. Hi, I'm Jonathan from the Buckler Lab.
Okay, thank you for Jonathan. Actually, it's also a good question. We did this experiment, and how much do you need for RNA-seq? Protoplast, you say, for RNA-seq? If
Yeah,
you...
my understanding is that most of your data comes from transformed protoplasts.
Yes,
Is that
yes. How much protoplast? I would say, for the cut-tag analysis, we're using 5,000, almost 5,000 protoplast. For RNA-seq, I would say maybe we're using the 200, how to say that? 200,000 protoplast. Yeah.
Oh.
More than that for each RNA-seq analysis.
let's see.
And we also test in root protoplast system. We did that experiment.
Oh,
Fair alarm.
that-
Okay, thank you.
Do we have any other questions or looks like he might be having a moment over at Cornell right now. Let's see. Well, guess if there are no more questions in the chat and nobody has their hands raised, well, it was a really interesting, very dense talk, but really interesting talk. I guess, shall we adjourn?
Yeah, sure. Thank you all.
Thank you.
Thank you.